Good, good day, good afternoon, good evening, depends where you are. Welcome to BioVerse episode 2021. And this one, we're gonna provide the highlights of the AACR, which has happened last week. And today is my great honor to have a very prestigious panel of speakers. Some of them are old friends. Actually, my fellow editors, Angus Liu, today joined me, as well as my old friend Jeff and Viraj. Who participated in last year's AACR. And also, I'm very happy to welcome our new friend, Dr. Ting Xu, who is the CEO and the founder of Alphamab Oncology. I particularly want to give a shout out to Viraj and Ting, because it's pretty late over there for Ting. It's past midnight in China. He's dialing from China, and Viraj from India. So this absolutely global event, actually. With that being said, for those who just dial in BioVerse the first time, let me do a little bit quick housekeeping, remind you or like introduce ourselves to you a little bit. I'll share introduction slides of ourselves. And Angus, you know the drill. Please give me a thumb up when you can see the slides, so I know the audience can see that as well. All right, perfect. Like I said, this is our 21st episode recap of AACR 2025 and the Asian Biotech Innovation Boom, which Viraj and team probably, and also Angus, can provide a lot of details. Who we are, first of all, BioVerse, where science sparks business in medicines. This is a monthly webinar co-produced by ourselves, ISWT Community, which is, we believe ourselves one of the most influential nonprofit for Asian biotech, as well as SAPA GP, which is a Sino American Pharmaceutical Association Great Philly chapter, not far away from where I sit, which is New Jersey. They're based in Philadelphia. And BioSpark, which is a Boston-based nonprofit focused on academic research and R&D in global biopharma biotech industry. With that being said, thank you, I would say, frequent attendees, and also we welcome the new attendees from around the world. And today, because there's a lot of data going to be discussed by our panels, so that they only represent their personal opinion. We do not provide or offer investment advice for your investment or your other activities. Really, just a personal opinion. This does not reflect the opinion from the ISWT community, SAPA, GP, or BioSpark either. Purely the opinion from the speakers. With that being said, now let's clear everything. It's my great pleasure to welcome the prestigious panel of speakers. What I would do, I would invite each one of the speakers give up to 1 minute self-introduction of yourself. Feel free to tell your company and what you want to talk about. So, but please keep that in mind. Limit everything in 1 minute. I'll start from my left. So, Dr. Ting Xu, would you please start?
Yes, thanks, Leo, and thanks everyone. So, basically, my name is Ting. I'm the founder and CEO of Alphamab and Alphamab Oncology. And I returned to China from Boston about 17 years ago. And currently, Alphamab Oncology is a commercial stage company focusing on IO and also ADCs. And actually, today I will take a chance just to go through some ADC I take from AACR and also share some of my personal opinion on the development and challenge of we can so-called next generation of ADCs. Thank you.
Thank you. Thank you, Ting. So, I look at the Ting's pipeline. I think you have a 7 clinical stage ADC right now. So definitely has a very knowledgeable about the ADC space. Next, my old friend, Jeff.
Thank you, Leon. Great pleasure and honor to be participating again. These are great events. So I'm EVP and expert advisor oncology at Lumanity and have a heritage going back through several companies. So I haven't had to change my seat despite name changes. Viraj and I work in a unit that focuses predominantly, at least in our corner of the unit, on helping early stage pre-POC biotech make key value inflecting decisions around their platforms, targets, indications, et cetera. So again, excited for the session today.
All right, Viraj.
Thanks, Leon. Absolute pleasure to be here on this webinar. As Jeff said, Viraj Parekh. I'm principal and oncology lead at Lumanity, as well as a partner at Clear Insights Bioconsulting, and again focused fully on oncology for the last 10 years, helping pre-POC and clinical stage companies navigate the dynamic oncology landscape. So really happy to be part of the discussion, and I think I'm going to focus on the RAS readouts at this year's AACR. Again, promising and interesting, but again, we can talk further about it in the course of the webinar. So again, happy to be on board.
All right, fantastic. Last but definitely not the least, my friend and a fellow editor and a fellow volunteer, Angus.
Hello, everybody. Glad to be here. I'm Angus Liu, Deputy Editor with Pierce Pharma. I currently lead our publications oncology coverage, and we're mostly focused on commercial in-market products wherever you are approved. So today I'm going to talk mostly about the commercial product, keytruda, and also some late-stage development from the immuno-oncology field. And Fierce recently we also launched a new newsletter, Fierce Oncology, which summarizes every story, most stories. We do cover a lot of stories. Most stories that we write across Fierce Pharma and Fierce Biotech, the two publications on oncology. And that I'm currently running that newsletter as well. So looking forward to today's webinar.
All right, fantastic. Let's dive right in. We have, now we have probably 80 minutes left, and let's make the best use of it. This is today what we're going to talk about. Ting, Jeff, Viraj, and Angus already mentioned a little bit. That's what we're gonna do. First, we really how to say one of the biggest topic, if not the biggest topic in AACR this year is IDC, and then we're gonna move to Jeff's multi-specific biologics, and then RAS, and finally finish with the late-stage immuno-oncology. So first, I'll pass the baton to Ting. So the floor is yours. Please, Ting, take over.
Great, thanks, Liang. So basically, it's kind of interesting. This is the first time I attend AACR. I always want to go, but never. And this time I take chance because I really want to present two of our own early stage pipeline. But basically, our ADCs build upon our 2-in-1 antibody. One is like PD-L1 integrin beta-6 ADC, and the other one EGFR-HER3 dual payload. But anyway, I found this AACR is pretty intense. Unfortunately, I don't have much time to attend all the seminars I want to go, but still get lots of opportunity to talk with people to get a feeling about what's going on in oncology research at this moment. And at first, I want to say that the ADC is kind of big part in AACR. We have about 233 abstracts focused on ADC. Again, we can see that China contribute quite a lot, about close to half ADC abstracts are from China. And then aside from kind of classical ADC, we start to see quite a bit bispecific ADC and also dual payload coming up. As of like dual payload, we saw 13 out of like 233. I believe next year we may get to see about 130, maybe even more dual payload. And actually for ADC, we know that it's still hot, but still in at least in China, people still start to question how long the heat will keep going. It's like the dawn or dusk of ADC, right? So that's today I'm going to go touch upon like what's my thought on that. Leon, next slide. So basically, currently the ADC into this stage after more than 30 years development, but actually the real time of development is really 10 years, maybe more than 10 years after A201 come in the picture, right? But as of today, so we are facing a challenge, like what's the next for ADC? The first is like, we're talking about lots of new target, like this year, AACR, we saw like CDH17 is coming up, like maybe more than 17 poster, preclinical, clinical around that. But do we really need a tumor-associated antigen or tumor-specific antigen? And how much, what's the expression level? This is a slide I take from a seminar. I don't even remember who, but this just give an example of HER2, right? So basically A201 and HER2 push the HER2 from really positive 3+ to ultra-low. And even they're testing negative breast cancer. So in a sense, every normal tissue have certain, breast tissue have certain level of HER2 expression. And so-called negative breast cancer cell still have some, but does this antigen really play a role in ADC or not? That's the first question. And then it put question on lots of with so-called tumor-specific antigen, like we can name a lot. Are these real necessary for develop next ADC? So that's first. Now next one. So the next is even more puzzling for me because we start see, lots of ADCs. We so-called target different target or antigen, but if with a similar payload, and they all show up in overlapping indications. Here, just an example, is just saying that in HER2 low breast cancer, we can see that in HER2 and Dato and also SG, and later in China, actually SKB264, all appear in the same space. If we take a deep dive into the data, we don't really see much differentiation in terms of efficacy. Duration may be slightly different, but it's cross-trial comparison. We cannot really see much. So in this case, if we look into NSCLC, particularly EGFR mutation, we can see even a lot more, like EGFR, CHOP2, HER3, you can name it, right? So, and SCLC, we can see BCMH3, DLL3, SEZ1, anything put into the with the same payload will show up no differentiation. And then it put another challenge. So what we are going to develop, how we can differentiate. The landscape is not only to compete with similar target, we have to cross target as a competition. So that just makes the development path very, very complicated. Leon, next one. So then we go back to the basic of ADCs, mode of action. Supposed to be simple, right? The antibody was labeled by our conjugate to a toxin and then got internalized, released there, and then tumor will develop resistance. This is took from ASCO education material from last year. So the resistance can become as a different way, like mutation, like pump out or some, But anyway, so what we are thinking about now is like, we look at ADC again, we can see that it's an antibody, we use a FAB as a recognition unit, that at this part, we can start to bispecific. We can do certain tags like epitag. We can target not only to the membrane surface antigen, we can target to like tumor microenvironment, right? And the payload-wise, to cytotoxic is expanding to anything else. But still, I don't really see something with particularly emerging other than toxins. Maybe some proteins or molecular glue may come as later as can be materialized. And still, we need to think more like what kind of other payload we can use to expand the ADC scope. And then linker. People start take a look at linker. Before, always think about, okay, GGFG, VC, VA, VCPAB, VA linker, whatever. And then now people think about how to increase the linker with like tandem cleavage with two enzymes to play, and then to adding more hydrophilic component to make ADC more stable and more kind of more specific. And then other than antibody-based ADC, we have like PDC with like lots of other component and come in play. And in this picture, and also missing one really important, actually two components. The first one is how we can do the conjugation. We always live with cysteine conjugation, but there are more. Like glycan conjugation is what we are focusing on. There are TG transglutamase based, there are saltase based. And so different conjugation also generate ADCs with different property. So that's critical. And then the other important things like a DAR, it's like we are talking about from Enhertu is DAR 8. But we can see here, like almost every DAR 8 ADCs associated with pretty much high toxicity, particularly hemotox. And then we can look at a few DAR 4, like Dato, right? And also we have our own experience with DAR 4 ADCs. If like we reduce payload to DAR 4, even MMAE is a very potent payload, but our MMAE ADCs, if they're DAR 4, they don't have that strong myelosuppression or hemotox. So the DAR, other than DAR 4, DAR 8, DAR 6, DAR 2, we still don't know what's a good number to put on toxins. So this is pretty important for like, particularly for dual payload, how we can do dual payload. Now we do like 1-to-1 ratio, like at 4+4, 2+2, whatever. So this is really going to be critical for make multiple payload ADCs. Then we move next one. Yeah, next slide, please. So actually, just to touch a little bit upon bispecific ADCs, Jeff is going to talk about more. We can see that there are a lot more bispecific ADC coming in, but there's still a caveat or problem is like, if we question what's the rationale behind this bispecific, why we put them together, I don't think many people really can give a strong rationale or supportive evidence. So, okay, we know that those two enzymes will work. We don't know. We did our bispecific ADC. The first one is biparatopic. Our simple hypothesis is biparatopic will increase internalization and increase the binding specificity. And our second one is CHOP2 HER3. We know that CHOP2 HER3 both working in NSCLC, we put them together. But we don't want to work on third one with similar simple approach. I don't think it's going to work. By adding, for example, Nectin-4 or CHOP2, my concern is not going to increase efficacy, but going to increase the toxicity. By putting EGFR or CHOP2, someone is doing that, we may run into a similar efficacy ADC, but with problem, a skin problem and stomatitis. So that's the problem and dangerous part. And all those things won't show up at preclinical development. So that's have to be proven in clinical trials. But that's just a note here. But from those bispecific, we can see that EGFR is come as the most common target for bispecifics. Here I would say that EGFR is the second most favorite target I would view next to HER2. Now next one, yeah. Now next slide. So basically BioVerse in China, we have already one EGFR ADC with DAR4 MMAE is approving, its BLA has been filed. But here in AACR this year, we noticed that there's a 6010. It's a EGFR monoclonal antibody with 8 payload with top 1 inhibitor. I don't know what's the exact structure, but this one I just show an example on the IO resistant NSCLC, non-EGF mutation. This one show very small number, like 3 out of 6 is OR, like 50%. Comparing the rest, we have to compare with like BL01D1 with like SKB264 and Dato, whatever. It stands out. The safety with 47 grade 3 above, mostly hemotox, is pretty decent. And they also, they have oral presentation on this one. They also showed very decent activity in third line EGF mutation in NSCLC and second line, with very few patients. very kind of impressive. Like in second line, they enrolled 10 patients, they saw like 9 PR. And in third line, they saw around 40% OR, So basically this again showing that EGFR can be a good target with good safety and can be even better comparing to 01D1. It's a wonderful drug with wonderful activity, but associated is more than 70% grade 3 toxicity. Leon, next one. So we talk about payloads, so they're like people are talking about a lot, like ISAC, like immune stimulator and RNI, whatever. But I view Biotech or molecular glue as the next most promising payload can be used in ADC as a mono payload or dual payload, right? So, but unfortunately, I don't see much presentation in this AACR. Hopefully, next year we'll see more promising data coming out. And then the right part is the dual payload. The reason we're thinking about dual payload, we always think that, okay, there could be synergy, but I would say that synergistic effect is always hard to chase, But then second could make more sense, overcome resistance, because if we view ADC as the majority is the chemo, the chemo always will become resistant, With the two toxin, the resistance were very hard to come up, And third one, overcome heterogeneity. Any solid tumor is heterogeneous population. Some could be sensitive to one payload, the other could be another one, could be top one or MMAE or something else. Like people are trying use like DDR together with top one inhibitor. But here, like just in this slide, most of the dual payload are based on chemical synthesis of branched linker payload. I see it's going to work to a certain extent, but it's going to be hard because you cannot change the ratio of different toxins. Maybe they have a few order of difference in terms of activity. And also those majority of the payload are very hydrophobic. Putting them together maybe can cause the product not that stable and limit its further development, right? So, and then just to say that it's going to be dual payload, if we think about it, it's going to be more difficult, more complicated to develop. And we have to carefully select what we're going to use to avoid the therapeutic index. It's for most of ADCs, the therapeutic window is already narrow. We don't want to make it even more a problem because if we consider individual people variation, the narrow window can cause big trouble later down the road for development. Leon, next one. So here, the 13 dual payload, it's covering lots of target and also some interesting combinations. And ours, Leon is nice put our 021 at the top. We are using EGFR HER3, like 2-in-1 antibody, and put DAR2 MMAE and DAR4 top one inhibitor. We saw like tolerance in animals is pretty good. And hopefully by the end of this year, we can start human trial to look at how well this dual payload will behave. We saw some others already entering clinic. There's one more not on this list. It's a CECM dual payload from Innovent. Maybe hopefully by the end of this year, we can already see some the therapeutic index coming up. Next one. So basically, another thing I want to see from AACR is a combo, ADC combo with IOs. It's not new because we know passive combo with keytruda already show really good data in first line. It's already approved. But here we can see like more trials because this one is basically the Biotech put huge effort in terms of use their PM8002. It's a PD-L1 VEGF bispecific in combination with the ADCs in their pipeline. The left panel is just showing that it's a CHOP2 ADC in combination with this bispecific in first-line ovarian cancer. The tolerance is decent. It didn't increase. And the OR is like at 54% in ovarian cancer. It's not first line, it's a PROC. So if we think about comparing to what Mirazo in FR-alpha overexpression ovarian cancer, this data look decent, and the safety it look acceptable. So, and also Biotech is trying to combine their HER2 ADCs, B7H3 and HER3. We'll see more data coming from. But the question I have here is like, we're talking about ADC as a base, combined with this bispecific PD-1 VEGF, or someone will try. We are trying to combine with PD-1 VEGF. Well, that much better than just a simple combine with approved PD-1 drug. You know, how much benefit we can gain from this so-called IO bispecific. So that's the question, you know, we will see. Next one. And then we can see that this is an interesting one. It's a Claudin 18.2 from Junshi in China. It's like, it's a antibody with a DAR4 MMAE. They tried in first-line gastric cancer combination with their PD-1 and Xelox chemo. Surprisingly, the tolerance coming out pretty good. The grade 3 above toxicity didn't increase comparing to chemo. And the efficacy in first line, they have OR of close to 80%. It's good, not numerically better than, if we say first line PD-1 plus Xelox will come to, depends on different trials, it's like 50 to 70%. Right. We will see how much the duration is just too short to see the duration and, you know, translate into the PFS. But it's just indicating that ADC can be combined with chemo in first-line setting. Okay. Then we will see more ADC into first-line, combined with IO, combined with chemo. Here, just say that someone saying that chemo-free is important. I used to believe we have developed something chemo-free, but I changed my mind. You know, if I view ADC as a chemo, as a major component, we may use ADC to replace certain component in standard regimen of chemo. For example, if we use top-line inhibitor ADC to replace irinotecan from, you know, warfarin, something like that, that may be still tolerable and will generate more response. And then use ADC alone, or ADC with IO as a maintenance, that could, you know, have some generate more clinical benefit. But again, so as of this stage, what we are asking for ADC is not only for efficacy, but safety will coming up as the most critical thing, because we start to see SKB264 in combination with oxytinib in first-line EGFR mutation. And Merck is trying to use SKB264 as a maintenance therapy after chemo induction, chemo and PD-1 induction, and then use their keytruda plus SKB264 as a maintenance. We are thinking about this like we are thinking about maybe 8 months, 9 months, even longer duration of treatment. In this case, safety, for me, from my personal opinion, comes even more important for next generation ADC development. Okay, I will stop here. Thank you. Yeah.
All right, fantastic. So also a quick housekeeping. So where's my face? Well, quick housekeeping. If you have any question, we do have a Q&A button. I believe somewhere in your screen, depends on which device are you using, like computer or, you know, a phone. But please put your question only in the Q&A so all the speakers can see it. And also I suggest you to upvote if you see some question already there. So I'll pick them because we have a limited time. With that being said, let's move on to the next topic. Jeff, floor is yours.
Great. Well, thank you. Thank you for that nice dive into ADCs, Ting. So I'm going to try to fly a bit over these slides and talk about some of the issues, even some of the business issues that kind of come out of the data out of AACR instead of diving specifically into a lot of the data. I do want to pick up on something that Ting said though, because it's very interesting, right? There are still people. Sometimes I've even said that I think it's referred to as, souped up chemo for referring to ADCs. And there still are safety and tolerability issues. And yes, in an ideal world, I think many people, oncologists, patients would like to be able to avoid any type of chemo and the safety issues associated with that. All right, maybe we will get there someday. And there certainly are people who are looking to obviate the use of, say, a backbone chemo regimen and create kind of a chemo-free. But until we get there, the ADCs I would consider a nice next step, right? And if we think about how long it took to get from the age of chemo, taxanes, platinums in the early 90s, before we had Herceptin, before we had kind of targeted agents. Obviously, we had early ADC with Mylotarg and then, of course, Kadcyla. So, that's a long ride before we get to the improvements that we're seeing and the rapid improvements we're seeing with ADCs over the past, 3-5 years, whether that's issues of different types of payloads or different types of linkers. Not to mention the issues that Ting talked about in terms of the multi-specificity or multi-payloads. And I'll touch on a little of those again. We'll just kind of emphasize some of those aspects. But I do want to go back to something that has struck me, and maybe we'll have a conversation. Hopefully we'll have time at the end because I'd love to hear from Ting, because one thing that struck me, it was from both a publication and also a triple meeting presentation by Professor Colombo of Zymeworks about how this is, an ADC is really a multivariate entity, right? And it's not always easy to rationally determine, the effects that you'll have between your linker, your DAR, your specific payload, where the things are linked on the antibody. Those all can lead to variability in behavior and therapeutic index. So it's, you can do a lot of rationalization, and certainly a lot of companies are being well funded for, coming up with innovative approaches to any one or multiple of those key elements of an ADC. And yet at the end of the day, I think it becomes very empirical. You need to do the tests in humans to really see what's going to happen in terms of the safety and the efficacy. So maybe hopefully we'll have time at the end to talk about that, Ting. But certainly one trend you can't ignore has been the ADCs. But also there's this broader trend ongoing, which kind of is encompassing of some of what Ting talked about on ADCs, and that is the idea of multi-specifics or multifunctionals. I keep going back to something that I think Dan Chen said once it was an IO360 meeting maybe, and certainly in conversations I've had with him, he continues to talk about kind of the engineering aspects of kind of an important wave of where oncology is going. And that includes, all sorts of engineering, certainly of biologics, aside from, other entities. So while there's been all this discussion and all the kind of abstracts and data that, for example, are represented here from the BioCentury article in terms of VEGF multispecifics, and they're not all PD-1 VEGF multispecifics. I think just the idea of this multifunctionality with the hope that that will improve on the agent's separate behavior, both individually and in combination, as well as conceivably display some unexpected activity that might lead to, maybe you can obviate the need for a chemo regimen depending on some of these combinations, and talking about physical combinations. And I think one of the things that, it's not in this table, but as I was kind of preparing for this, I thought was really interesting is the Shantibody therapeutics, so more innovation coming out of China. And this is like a tri-specific, right? So it adds the CTLA-4 modulation into the PD-1 VEGF. And that's really interesting because, I was curious, we did some of our first work in bispecifics probably around the 2010s for F-Star and a few other companies. A lot of activity then. They haven't seen the light of day yet as an entity. I think they've gone through a number of restructurings. When we did a large look at the bispecific landscape for Genentech in about 2016, 2017, at that point, I think there was, other than the clinical stage cell redirecting bispecifics, I think there was only one clinical stage true bispecific that was immuno-oncology, meaning two IO relevant targets. And I don't even think at that point, maybe there were some HER2 and IO combinations, but it would have been very early days. You fast forward now, almost a decade later, and we're still kind of trying to move forward with those. And, everyone's hoping, and the data right now would seem to be suggestive that we will have this major breakthrough with whether it's Evo from Akeso and Summit or something else, that, this will be a kind of a major game changer. And it's not to say there haven't been other, obviously we can't forget Rybrevant, its approval, but I think the progress of the bispecifics has had its ups and downs, just like ADCs have. And we have to bear in mind that, these are complex molecules to develop, to manufacture, to test. And sometimes you do get unexpected behavior, like we seem to be seeing with the VEGF PD-1s, which, when they were used separately as, say, a checkpoint inhibitor and Avastin, or a checkpoint inhibitor and MTKIs that, modulated angiogenesis, there are some approvals, but by and large, either the efficacy or the tolerability or both were problematic. So just interesting to kind of step back and see the evolution in the space. Next slide, please, Leon. So again, part of this kind of what I've called the dance craze with multispecifics, is really being fostered, in the past, 18 months, I guess, or so, because of the Akeso and Summit data. And of course, not that the street's a surrogate that we want to overly focus on, but it has been interesting to see how the street and the industry has reacted as the data have come out, both from Akeso and the China studies, as well as, from kind of Summit. And even the more recent pre-ACR release has led to, interesting responses and debates around how statistically significant it should or shouldn't be, given that it was, before, the actual scheduled interim readout. And, but I think at the end of the day, whether you take I think the Lancet publication, someone correct me if I'm wrong, was like almost 50% reduction in risk. So right now with this interim readout, it's almost half that. Hopefully it doesn't go half again, right? That's what we saw with NKTR214, the Nectar-IL-2 agent, right? Great data, and then each time more data matured as that progressed through, it kept getting cut in half until ultimately, it was not meaningful, and we know where that story ended up. So I'm hoping more for patients' sake than anyone else's sake that that this story holds true for these PD-1 VEGF agents. Next slide, please, Leon. And of course, when talking about the space, I think aside from the perhaps unanticipated value of literally combining these two together in a single molecule, and that goes back to some of my first comments about the history of bispecifics, there were people along the way, including F-Star, who I mentioned too, who started to say, oh, we're seeing different behavior, and therefore, these are, if it's a PD-1 with something else on the other end, we're seeing conditional behavior, right? So in other words, that antibody is being trapped or localized at the tumor or in the TME because of high PDL expression or something like that. And that's interesting, although, that hasn't by and large borne out, but clearly something's different happening here. And so that's led to this big sea change. And of course, there was the big move that BioNTech did with like around $800 million, I think, acquisition of Bioethis. And then of course, Merck deal with Lanova. So, as is common in the industry, there's suddenly this rush in. And of course, while we will focus and should focus on the value that these may bring to patients, we can all understand from a financial standpoint why these companies are rushing in to protect these huge, multi-tens of billions of dollar checkpoint franchises. And of course, for BioNTech, they're not protecting that. Merck obviously is, and others. But I think it's been fascinating to see BioNTech evolve from basically an mRNA company, but they've long had, oncology as a focus. So it's not like they suddenly came into oncology after having, had their big win in COVID. It's long been an interest of theirs. They still have a lot going on with mRNA-based therapeutics, including, of course, vaccines. But this move into, classic, we'll call it soluble biologics, no longer kind of mRNA programs, I think is really, really interesting. And at the same time that they're trying to compete now with this bispecific, they're also doing what Ting also alluded to, combining with ADCs. So they would have their own kind of control of that regimen, whether it's the TROP2 combination or others. There are a number of them listed here on the bottom right that they've done kind of combinations with, including, B7H3, which a lot of people are pursuing, and obviously HER2, which, just about everyone and their mother is pursuing. But interesting early data, combination data, as it said, similar to kind of what you saw with the Genmab folate receptor targeting ADC. So it'll be really interesting to see how kind of the data continue to mature in this space. Next slide, please. And of course, as ADCs continue to innovate, and this, we came across the same slide, Ting and I, from Beacon, Hanson Wade people that put out, great data, and databases are really good. But it's been interesting to see, kind of the evolution now of these bispecific ADCs, and bispecific predominantly really meaning, dual targeting. As I make a joke of in the title, I've seen now people calling them BS ADCs, which I don't think is a good name if you know what the acronym BS means. But I think it is really interesting to think about, whether these bring additional specificity and improve the therapeutic index, whether they account for resistance mechanism of shedding of one or the other or downregulation of those targets, whether they lead to some interesting unanticipated cooperative effects because of those targets. And of course, another thing that I'm not sure is talked about as much is that when you have something that is a bispecific, it does lead to potential broad franchise development, because sure, you can go for tumors that have high expression of both, but you could conceivably go for tumors that have high expression of one or lower expression of one or both, which means that you've kind of multiplied at least by two kind of the patient populations or that you could conceivably go after, assuming that those targets are not completely redundant, in their expression level. And of course, I do want to shout out to, one of our big clients, Gensai, who has had a number of their bispecifics that were presented at AACR, targeting, B7H3, which I said before is a very, kind of common target now, of course, FR, and that's a biparatropic agent, and EGFR HER2. So, and so, of course, there's the dual targeting, and then as Ting also mentioned, so next slide, please, Leon.
By the way, Jeff, just let you know, your client's VP of BD actually is in the audience.
Oh, okay.
He would appreciate your shout out.
All right, great. Good to hear. Yeah, I'll take a look. So, and then of course, the idea of kind of the dual payload, which I think Ting kind of really noted, and the questions will be there in terms of what do you get versus what might you lose. Clearly, the idea of kind of non-overlapping, if not orthogonal, mechanisms of action is important. That obviates potentially, the mechanisms of resistance. It also serves conceivably as a way also to block someone who might be developing a follow-on, right? Because we have these discussions all the time. Oh, if you're going to develop a TOP1 inhibitor-based ADC, then you're going to have to think carefully about where you're going to develop it. You don't go after one that's already approved and used, and which you presumptively have resistance if you're going after, next line patients who have progressed. Whereas, you put in another payload, right at the beginning, you may be able to either avoid that resistance in the first place, or certainly cover yourself for potentially going in after people that might have had a single payload before. So again, you kind of magnify your potential applicability, but you also deal with issues of tumor heterogeneity, possibly low expression levels, and so on. So next slide, please.
If I was wondering, can I just make an observation?
Yeah,
on the
please
last
jump
slide?
in, Viraj. Yeah.
Yeah, I was just, I mean, it's interesting, like Ting talked about it as well. Leon, if you go to the prior slide, I mean, some of these are using traditional, tubulin and topo-1 combinations where potentially you can target different things and, you're overcoming some of the resistant mechanisms to the payload. But I mean, some of these are also very interesting where they're using, innate TLR7, 8
That's a good,
agonists. Also PARP1 inhibitors. Some of that, I mean, RNA pol-2 is not a validated ADC payload yet, because the idea is that you can target both DNA and RNA, and then potentially, do a much better job at, with the TROP2 targeted agent. So I think it's very interesting, but to Ting's point, very still early to understand how these payloads are going to play together. Yes, individually, a TLR7 and 8 might make a lot of sense with, let's say, a chemo payload that is, causing ICD, causing, some innate activation. But when they're all together in the same compound, attacking potentially the same cells, of course, there's some bystander effect and otherwise, but it's going to be, like you said, empirically, you have to then assess and evaluate, because even at with the AI and all of the other stuff to understand, and before you actually put this into cells, mice, humans, it's going to be difficult to understand exactly how these
absolutely.
play
And
together. Yeah.
thank you for bringing in. And I did want to mention, in particular, the idea of the combination of, even in a single molecule, let alone separately, which would still be relevant, of course, just like the PADCEV keytruda combination that Ting mentioned, the idea of kind of having the cell killing and the immunogenic cell death linked in the same molecule with something that is going to take advantage of, in some way, either immunostimulatory or countering the immunosuppressive aspect, I think is a very appealing concept. I don't know if it's, something innately pun intended, a little bit more than, say, a dual cytotoxic payload, just because of concerns that have been raised about the tolerability of that. But, we'll see. Hopefully there'll be space for all of those. But thank you, Viraj. To the next slide. So I did want to kind of talk about a little bit about, so these dual payloads, I think I already mentioned some of the kind of advantages, which Ting also did, but I thought I'd just show a snapshot of another of our clients, Sutro, and give them a shout out, although they did have to go through a little bit of reorg and kill their lead program. The TF program looks particularly interesting with their novel payload, but also they presented data, which is what this slide's about, of those dual platform, which includes, as Viraj said, like combining a PARP with a kind of a more classic cytotoxic payload. So again, interesting places, a lot of innovation occurring, a lot of it very early right now. So that empiricism, meaning we really need to see what will happen in humans, is going to be critical. All right, I think I have one more slide, maybe two. Trying to kind of now branch out a little bit. So multifunctionals was where I started. We spent a lot of time on VEGF related, and then ADCs with dual targets and so on. So that idea of multifunctionality, this was one thing that kind of stood out, that was interesting to me, was Marengo's, for a variety of reasons. One, this idea of kind of targeting the V beta as a way to, kind of be a TCR activator, but also having this fuse to IL-2, just given the history of IL-2, which I already mentioned before about Nectar-214. And they had, promising data. It was kind of presented here in terms of kind of the disease control rate, et cetera. It seemed to be relatively well tolerated. When you kind of look at the data, they had evidence of some memory effect. So kind of this idea of, and other people are doing this now, right? They are taking things like T-cell engagers and adding other elements to those to kind of soup them up. I think even some T-cell engagers that may be ADCs now, correct me if I'm wrong, Ting. So I think, the kind of the coalescing and the cross disciplinary combinations that people are looking at now in terms of modulating IO, having toxic effects from some type of payload. Of course, the next generation payloads like PROTACs, but also IO agents. So just lots of interesting stuff going on. A lot of it around engineering of biologics. I mean, mostly we've been talking about antibodies or fusion proteins. There are obviously things like the DARPins and the radio-DARPins that Molecular Partners is working on, or other companies that are working on nanobodies or other macrocyclic delivered ADCs. We didn't even talk about those, right? So it's not all biologic delivered conjugates, but anyway, so is that my last slide? I don't even remember. It is. Okay, good. You're up, Viraj. Let's switch to small molecules now.
Yeah, before that, I want to remind the audience that for the sake of time, obviously time is not our friends, unfortunately. So please put your question in the Q&A. We're going to dedicate quite a few minutes. By the end of the, the panelists' presentation, they're going to put all the questions together. In the meantime, take a review of all the previous questions. Probably someone already asked a similar question you want to ask. Just give them a vote. So we're going to say them on the very top of the question list. With that being said, Viraj, floor is yours.
Thanks, Leon. So yeah, moving to more targeted therapies, and just trying to set the context. We've been trying to probably target RAS for the last 40 years, right? I mean, until we discovered the druggable pocket in KRAS about 10 years ago, we didn't really make that much headway. But since then, there's now dozens of KRAS, pan-RAS approaches that are in development, and also we have now 2 approvals in adagrasib and sotorasib, now not only as monotherapy in non-small cell, but also in combination with EGFR targeted agents in CRC. But as you I mean, we've come a long way, and of course it is very interesting because RAS, specifically KRAS, is known to be drive so many tumors. Many of them are commercially very attractive, also very high unmet need in non-small cell, PDAC, CRC, among some of these, melanoma, thyroid. So again, a very, it's going to remain an active space, I think, for the foreseeable future. And it does come with a lot of nuances and dynamics because historically, yes, difficult to target, but also as we've developed agents, have some approvals, we now are understanding the very nuanced, landscape of, different alterations, evolving resistance mechanisms. Do we target RAS off? Do we target RAS on? Do we use degraders? Do we use molecular glues? Do we use small molecules? Can we use IO approaches like vaccines to target RAS? So again, all of this is kind of now coming all together slowly, but surely incrementally going in the right direction. Leon, next slide, please. So again, like talked about, so we made quite a few, advances. We now have, late stage pan-RAS, RAS-on targeting agents by, like Revolution Medicines 6236. And again, using modalities beyond small molecules. But again, there's still significant variability in activity, right? So if you look at non-small cell, you have single agent activity of 40%, responses and a PSS of 7 months. And if you look at, CRC, again, G12C driven to some extent for, 20% of patients, but still, you needed a EGFR backbone to then get a 30% response rate. So again, on its own, the G12Cs weren't that clinically relevant in CRC as they were in non-small cell. Again, underlying that this is very nuanced and, the heterogeneity, not just within the tumor, but across tumor types is not something that one size fit all kind of approach is going to make the cut, right? So, and you do see, some of these, advances like, the pan-KRAS is doing, PDAC, which is historically, a difficult tumor to treat. And again, we have a cancer vaccine that finished its phase 2 in KRAS-driven tumors and now is going for its phase 3 in locally advanced PDAC in the adjuvant setting. So it's not only in the metastatic settings, but we're also looking at earlier stage settings with the vaccine approaches. But with all of this, on the right that you see, there is so much new data coming out on resistance mechanisms. This is just a snapshot of KRAS G12C. So there is, of course, the RTK hyperactivations. There is RAS amplifications. There is, MEK, ERK, alterations. There is the PI3K. There is adjunct pathways of HER2 activations, HER3s, and the SRC pathway activations. So all of these are playing a role in patients progressing eventually on these KRAS, G12C, and other inhibitors, right? And another, kind of evolving thing that we're seeing now, of course, KRAS G12C, of course, is a relatively clean target, but of course you do see, basic level of toxicities with these first generation agents. But there's additional kind of new data coming out is liver toxicities that we're seeing with these KRAS G12C inhibitors in patients that have progressed on checkpoint inhibitors. So again, considering all of this, there definitely is a unmet need across the board on developing better, RAS targeted agents. Next slide, please. So there is some, new data about, I think there were about 7 or 8 clinical presentations on the RAS targeted programs, and probably 70 or 80 preclinical, readouts here. But I think one of the more encouraging ones was the RevMed's G12D targeted program. So this is a RAS on G12D inhibitor. They have, 18 patient data for non-small cell with, with a response rate of 61%. So again, this is relapse refractory patients post checkpoint inhibitors. Typically would get docetaxel ramucirumab, which is probably at 20%. And even if you compare it to other in-class competition, like, Astellas's G12D degrader, which is probably at 20-25% response rate, and also others which are, RAS off, which have about 15-20%. And again, even comparing to Revolutionary Medicines' pan-RAS inhibitor, which had initial data in this population at 40%. So again, clearly within this population, which is sizable still in non-small cell, it's a really encouraging response rate of 60% plus. Again, not as great as you would expect for EGFR, osimertinib and the ALK selectinibs are at 80%, but comparing where adagrasib and sotorasib were in G12Cs at 30-40%, this is certainly a significant step up. If you look at the safety data, it does look like it's reasonably safe. Again, it's only 18 patients, so we need to get more mature data. But kind of linking some of that liver tox that was seen in some of the G12Cs, you don't see that with this. So if you look at, there is slight increase in one patient in the ALT, but overall the AST and other liver-related toxicities are grade 1, 2. Overall, no real discontinuations, very little disruption. So, it does look like overall it is reasonably well tolerated. Again, it might be from the fact that it is selective, it is RAS on, and, it is fairly potent. So again, the idea probably for them is, monotherapy initially, and then combination with not just RevMed's pan-RAS, which there is preclinical data, which we'll talk about, in the next few slides, but also, standard of care regimens that, other adagrasib and sotorasib agents are already doing in earlier lines, right? Eventually, I think the goal for this agent is going to go into, either first line metastatic, where you have, more patients, or, locally advanced stage 2-3 patients, like osimertinib and other targetable, AGAs are going into. So I think, this is probably one of the best data readouts that the RAS targeted programs have seen in AACR this year. So next slide, please. I think another interesting one, again, is, again, D3 Bios, which is a China-based biotech. They have a bunch of KRAS programs and bispecifics, but this is one of the lead programs, the next-gen G12C inhibitor. I think it's interesting because they've got data across the board for a non-small cell CRC and PDAC. Again, it's initial, but in both naive and resistant population. So again, if you look at the naive KRAS G12C naive population, they have an overall response rate of 70% plus. And again, this is in relapse refractory, non-small cell, high unmet need populations, even in CRC and PDAC, but has not been treated with first generation KRAS G12C. But if you look at non-small cell, it's about 67%, CRC 88, and PDAC is 75. All of this is well above where some of these first generation agents are, of course, but
Can I ask-
also, yeah, go ahead, Jeff. Sorry.
Can I ask a
Go ahead.
question, Viraj? So
Mhm.
that's unanticipated. Do they give an explanation mechanistically for why that is, right? Because normally we've seen the decrease in activity as you go from lung CRC to PDAC.
Yeah, yep, exactly. So I think one of the reasons was enhanced target engagement, which they probably, and also overcoming some growth factor induced nucleotide exchange. So basically what they're seeing is this is a RAS off. So they're basically inhibiting and for long enough the GDP not converting to GTP. So essentially it's probably a more potent compared to some of the other, but you do see some of this data also with Roche's G12C. So it's not unheard of. I mean, in the non-small cell, and Roche has seen some of this data. But you're right. I mean, the data in CRC, again, it's only 9 patients and 4 patients with PDAC, but that was surprising for me too. I mean, it's higher than non-small cell and almost 90% in CRC. So
Yeah, it's just
and
that it's,
yeah.
I know previously in discussions, we would talk about, oh, this is a
Mhm.
reflection of the tumor context, right? And the
Correct.
different signaling pathways between lung and CRC and PDAC, as opposed to something tied to the target or a way to drug that target per se. But this would seem to counter that idea.
Yep, yep. I mean, it does, yeah. So I mean, but again, it's initial. So like you said, I mean, as we get to more patients, I think they're planning to accrue at least 20 patients in each of these cohorts. So we will see. But yeah, initially this data does look, definitely above or where the current landscape is. And if you again go on the right, some of the safety data, and I think 600 milligram is where they're focused on their phase, dose expansion studies. And again, no real discontinuations, no serious treatment-related AEs. Again, some expected GI, nausea, diarrhea, some liver tox, but nothing really standing out. So again, relatively safe. And I think the really more exciting piece of data is as we think about sequencing and when we're thinking about resistance mechanisms and potentially, we are going to get to a point where you're going to probably use multiple KRAS directed inhibitors in sequence to make sure, we get that extended, durability of response across these targeted therapies. They did extend the study to 20 patients in non-small cell who had been prior treated and progressed on first gen G12C inhibitors. Mainly, I think it was sotorasib and adagrasib, but some other ones as well. But it did see, an ORR of 30% and a DCR of 80%. So again, this is potentially third line plus non-small cell patients who are pretty beat up because they're probably platinum front line, G12C inhibitor second line, and then a third line. Basically, this is post even docetaxel ramucirumab in most patients. So we're basically expecting response rates of 10, 15% and a PFS of 2 to 3 months in this population. And seeing a 30% response rate, again, it's early 20 patients, but I think this again is key here as we think about development of the KRAS and RAS space is, the identification, understanding, and then how do we target, the resistance mechanism, either via sequencing, combination strategies, and, potential different modalities like degraders, molecular glues, which can potentially be more effective than the traditional small molecule inhibitor approaches. Next slide, please.
Actually, a quick question for you. So since you are such
Sure.
an expert of a RAS pathway, obviously, I think from Wall Street point of view, the most interesting one is the Revolution Pan-RAS. I think it's a RAS-on. And there's a little bit skin toxicity. Do you think that's a kind of on target of tumor toxicity? And also, I believe they reduce their dose when they switch from one indication to another. What would you think the strategy if someone can develop next generation pan-RAS inhibitor? What should they do to, let's say, have a better drug compared to what a Revolution Medicine already have in phase 3?
So I think some of the other pan-RAS strategies are trying to include. So I think some of them are just going pan-KRAS, and again, it could be pan-KRAS. Strategies like Revolutionary Medicine is doing, but also peptide vaccine and others. I think a more focused approach because I don't think some of this activity of they're basically Revolutionary Medicine with their 6236 is basically value propositions not only KRAS but NRAS and HRAS too to some extent, But I mean, that's not really probably adding to too much activity, especially in the patient populations they're looking at. So I mean, to be frank, it's going to be definitely a safety play, because even with so we have now sub analyses that they did on patients that progressed on the pan-RAS, which was actually presented as well, which I've not covered, but I was going to kind of do a voiceover, which great point, Leon, that they did see most of them progress even with the pan-RAS, and they did a ctDNA analysis and saw multiple resistance mechanisms like the RTK hyperactivation, RAS amplification, a lot of adjunct pathways, downstream activations and alterations. So the idea is now, with that, they did a lot of preclinical subsequent data with this agent that we just talked about, the G12D, and they did see significantly better activity in G12D mutated patients with a pan-RAS and the G12D that we just talked about in a G12D resistant patient population. Additionally, they also saw HER2 amplification. So they actually combined with Enhertu, saw a significant, kind of additive, well, let's call it, added effect with the, Enhertu. And so I think eventually, even with the pan approaches, it's going to be a combination play. And that's where I think the safety aspects are going to be really critical, because when you combine a pan RAS with, let's say an HER2 or a G12D or potentially another adjunct, activation, or let's say an ERK or a PI3K or AKT, you are expecting, added toxicities. And I think, especially in late metastatic disease, even in frontline, there's, they're pretty, they're metastatic patients, so you don't have that ridiculously large therapeutic window. So I think you're right. I mean, one aspect of where the other pan-RAS agents could differentiate themselves is the safety, and the second is the combination strategy. I think that is evolving, because I think understanding the resistance mechanisms, because what they've also seen is RAS on resistance mechanisms are differing and deviating from RAS off base resistance mechanisms, which is a very a nuance that they have to then consider for inhibitors that are targeting RAS off GDP versus the RAS on that revolutionary medicine is doing. So a lot of things to consider, but yeah, one aspect would be safety and clin dev combination strategy.
Yeah, I have one comment on that, because like pan-RAS basically is a molecular glue with a cyclophilin A, right? So it's
Yes.
actually someone has already trying to use pan-RAS inhibitor as a payload for ADC. I don't know, is that going to work out or not, because the scale-wise doesn't seem fit so far, but it could be a direction to solve the safety and efficacy issue.
Yeah, actually, there are other absolutely thing, and I think we'll talk about. I think if you go to the next slide, there is these in yep, these MD Anderson has a GRAS G12D siRNA based exosome delivery platform, which is looking at relapse refractory PDAC patients, again early data. But again, they did see, again, downregulation of, tumor G12D DNA, suppression of downstream phosphorylation of ERK, increased CD8 T cell infiltration. So again, I think with, like with the ADC, I think the KRAS, and again, adagrasib is doing it already in the clinic, is combining with, keytruda, combining with chemo. So I think the idea here is taking an IO-based, or like exosome-based approach in this case, and then also tying it to potential immune effects, which can then potentiate or additional combination strategies like they're talking about here with immune checkpoint therapy, like PD-1s or others, in either PDAC or other models. So I think you're right. I mean, they did see 33% stable disease. Again, that's not much, but still in a relapse refractory metastatic population, it is something. Again, very early, and I don't think they're expecting this to be monotherapy in any way. But I think the idea is that they wanted to get some sense of, is there any activity with this kind of exosome siRNA based approach that's not small molecule, and also showing some immune correlate data to which then they can potentially talk about ICI or other immune combinations.
Stopping
again,
myself from asking any
yeah.
questions or making comments on the vaccine approach, because that's something that, we've done work on,
Yep.
going back many, many years, but I want to make sure we leave time for Angus too. I'm sure Leon does
Yeah,
as
yeah.
well.
I'm going to be relatively quick here. So again, yeah, on the right is again a KRAS vaccine approach. Again, 6 most common KRAS approaches. This is combination with IO in third line plus again high unmet need MSS, CRC. Reasonable safety. They did get a response rate of 10%, which is again not that great. DCR is pretty reasonable at 42%. A PFS of 4 months and a median OS of 24 months. Again, I was really surprised with that median OS, and I'm still trying to dig a little bit deeper. I think it's probably taken from the initial first line treatment all the way to now, because because I don't think even first line MSS patients have a median OS of more typically 24 to 30 months. So I think this is probably that, but still there's some activity there. This is all of course non chemo based. Again, in this line, it's all, regorafenib or Lonsurf, which is 1% response rate or whatever, right? So again, very early data, but again, these are non-small molecule approaches that are, gaining some traction and have some semblance of activity in these KRAS patients. Again, one last slide, and then I'll stop. Lots of preclinical data, as I talked about. I think most of it is trying different combinations and understanding how they can de-risk first in human studies based on as much preclinical combination data they can get either with MEK, HER2s, HER3s, SOS1s, SHP2s, SARCs, all of that is in there. Again, nothing really stands out, but you have a couple like a pan-RAS molecular glue. Again, 8 to 21 times apparently the cyclophilin A binding versus the RevMed's pan-RAS. Did see some strong data both as monotherapy across non-small cell CRC and PDAC models. Again, there is Ryzen Pharma's PROTAC based approaches. So again, we're going to degraders now beyond the again G12C, G12Ds and Vs. Again, so significant suppression again, but very early in vivo data. But again, these are all the upcoming different approaches beyond the traditional small molecules. And again, on the right is Revolutionary Medicines, new RAS on G12V. Again, much more potent, much more selective, and they're saying, again, with less off-target effects as some of the MEK inhibitors have. So I'm going to stop there. Again, I know that, we're running out of time. So thanks, guys.
Okay, Angus, save the best for the last, I guess. And bring back the glory of immuno-oncology, please.
Oh my God,
if you could.
I highly doubt that. No, AACR is typically very research early stage based. But we do sometimes have some of these later stage immuno-oncology, later stage stuff. Next slide, please. So the positive news here, even though we're kind of running short of that in immuno-oncology in general, is from Keytruda in this perioperative head and neck cancer space. This is from the phase 3 Keynote 689 study in resectable locally advanced head and neck squamous cell carcinoma. The design was quite kind of universal with perioperative these days. You add Keytruda on both sides of surgery to kind of the standard of care, which is a radiotherapy with or without chemotherapy in this case in the adjuvant setting. So here Merck was saying that this is kind of the first positive trial in this setting in more than 20 years. So they believe, and AACR has this press release to us as well, saying that this is going to be a new standard of care in this setting. The primary endpoint is event-free survival. There's a 27% improvement in all comers, and the median EFS, as stated here, 52 months versus 30 months. Quite some improvement here. So the co-PI, principal investigator, saying that this is going to be a new standard of care, and FDA has a priority review, which is going to be late next month. But there are a few things I think that are kind of relatively universal to some of the PD-1s. Next slide, please. next slide. Yeah, there are mainly three things. First of all is the PD-L1 active population, because the 27% improvement covers all comers. And this one is not very impressive compared with some of Keytruda's other performance in the perioperative setting. In non-small cell lung cancer, for example, there's a 42% improvement in KEYNOTE-671. This is, again, event-free survival. And the size of the benefit kind of decline as we include patients with lower expressions of PD-L1. Like you started from this kind of how their statistical analysis was designed. In first analysis, analyzed the CPS at least 10 population. The improvement was 34%. And once you include the CPS 1 population, it declined to 30%. And the all population is 27%. So there's definitely a question about the PD-L1 negative population. Merck's response was like, even if there were just 2 to 3% improvement on EFS, the absolute difference, we have seen some uptick in there as well. And she also pointed to the median EFS, as I mentioned, that was kind of a big improvement from like 30 months something to over 50 months. So the median EFS was really kind of impressive. And also the principal investigator said that only 5% is a very small population for the PD-L1 negative population. So it's kind of hard to really understand, given the small population, what really happened in that patient group. So he was like, as a whole, this should be still be approved or should be an option for all patients, regardless of PD-L1 expression. The second question has been also around for some time since that ODAC meeting last year on Imfinzi on the perioperative design in non-small cell lung cancer, which is, do both neoadjuvant and adjuvant Keytruda contributed to the final effect? This was relevant because adjuvant Tecentriq basically failed without neoadjuvant. That is, adjuvant Tecentriq failed in this study, INVOKE-010. So we looked at we tried to, of course, the study was not designed to answer the question of contribution. So we tried to find some traces from different data points. One of them is a key secondary endpoint, which is major PCR, which was evaluated right after neoadjuvant treatment. So there is a 9.4% difference for Keytruda versus control on major PCR. So 9.4, Merck even acknowledged that this is a very small number. But because how that got translated into like a 27% EFS, you started to wonder, did adjuvant kind of contribute a little there, whether you really needed that to expand from a 9.4 neoadjuvant improvement to a 27% EFS improvement. And as to whether neoadjuvant really contributed, that, like I said, the 9.4 was a fairly small number. So Merck and the investigator pointed to how the use of chemotherapy in the adjuvant setting. So basically, during the adjuvant phase, Keytruda patients in the Keytruda arm used fewer patients in the Keytruda arm used the chemotherapy versus the control patients did. So they were pointing to this as saying like, you reduce neoadjuvant treatment of Keytruda, reduce the need for chemotherapy, which is a positive for patients if you don't have to go through chemo, obviously. So, like I said at the beginning, FDA doesn't really like perioperative design after that ODAC meeting. But I think the consensus among the industry is that the agency likely won't retrospectively punish existing studies, and we have seen, because that ODAC was about Imfinzi, about AstraZeneca, even after that, the FDA has already approved that perioperative non-small cell lung cancer indication for Imfinzi, and also another muscle-invasive bladder cancer indication for Imfinzi as well. So this likely won't affect this design from an FDA perspective. But for future trials, you definitely want to see, the FDA definitely want to see contribution from each phase of treatment. Next slide, please. So the third thing is overall survival. Right now, the CPS, the first analysis was done in CPS, again, 10 population. There, the statistical significance bar has not been reached, but given how this curve separated, this is likely not going to be a problem with the FDA. But again, the question is, what's PD-L1 negative population is going to do? How that population will benefit, but that's likely, again, given the small population, we probably won't know for sure. So next slide, please. Now on the negative, which is like everybody has been waiting for, and we've not already known before the meeting. This is Roche's TIGIT study, tiragolumab, the Skyscraper-01 study in first-line PD-L1 high non-small cell lung cancer. Next slide, please, Leon. It has failed on both PFS and OS, and specifically, people were kind of hoping that overall survival, because the positive sign from the leaked interim analysis that we saw in August 2023, there's 19% looked very promising. We were hoping that it's going to expand, and given Roche management's comment that they were hoping that they were putting more kind of eggs into the final analysis. But unfortunately, this didn't pan out. The OS benefit deteriorated from 19% to 13%, and the p-value, it was not even close to the significance boundary. So this was kind of an absolute failure. The median overall survival improvement was also moderate, only like 6 months from the combination of Tecentriq plus TIGIT versus TIGIT alone. So this kind of raised a question: is this TIGIT's swan song? Is this the last one? I say almost, because there are still a few things that are ongoing. Because Roche has right now, based on their Q1 earnings, they have 2 phase 3s ongoing for TIGIT. One is in first-line stage 3 non-small cell lung cancer, and the more promising one that I personally think there might be a chance is this first-line liver cancer indication. It has shown some very promising phase 1/2 data. The overall response rate was 42% versus 11%, like Tecentriq TIGIT combo versus Tecentriq alone. So this HCC is, I think, TIGIT, Tecentriq, and Avastin combination, 3-component combination versus Tecentriq and Avastin. So the median PFS, again, is also some kind of very promising difference you see there. So this one might be their shot at getting something out for TIGIT. And we know that several other companies have already dropped out of TIGIT, but there's still 3 remaining players: AstraZeneca, Compugen, they have one, Gilead and Akers, and GSK, Ateos. They still have TIGIT programs going on. The thing, I talked to Lyrics analyst Dana Gray-Bosch about this. She has this theory about FC competent, whether you are FC competent or FC silent, because if you have, given that TIGIT, Roche program, and also Merck, Beijing, and their programs are FC competent drugs, they come with higher toxicity. But I think there is a discussion during AACR that questions like how much really the higher toxicity, how much did that really matter to the lower efficacy? You know, when we think about higher toxicity, is less time on treatment and higher discontinuation and shorter duration of treatment and everything. But there is a question like how much that really contributed to the lackluster efficacy that we've seen. On the other hand, we have FC silent. Some of those drugs, I think Gilead, Akers, I may be wrong, might be one. These ones have better tolerability. So the argument is that they may have a better chance at combinations together. And there's another approach which is being taken by GSK, which is like, let's just step on the gas. Let's just increase efficacy so much so that even the toxicity and everything that won't matter. You can just trump that. So they have PD-1 Jemperli, which they have already shown in the phase 2 PERLA study, which is kind of pointed to that it being a more potent PD-1 than Keytruda, because they use a higher dose of PD-1. So they're just doing this. Let's just ramp up the efficacy part so much so that the more toxicity. And we've seen that, I think, with their phase 2 non-small cell lung cancer disclosure. Was it at ESMO or somewhere else? Like, kind of you see a very high efficacy, but investors were kind of got a little bit worried about the tolerability, the side effects were kind of a little bit alarming, but GSK still like kind of pushed forward into phase 3. So I think their rationale is just basically, let's just crank up the efficacy and see what happens. And next slide is just
actually before next slides, quick kind of a question for you, Angus, and also to Jeff and Viraj, since you guys are doing strategy for big pharma all the time. So obviously the indication are going after solid tumor, particularly lung cancer, is so competitive, right? That's changing all the time. Let's assume this get approved. Based on the current data, the benefit going to be, I would say, very modest. Now we know there are so many other things ongoing, right? You saw like, Pfizer actually working with the Summit, right? So then we already know PD-1 plus precision chemo. That's how many people call ADC, have a lot of improvement. How would this pan out actually? Yes, you get approved and like 2 or 3 years have a much better drug on the market. How would that bring commercial value to those companies who are still pushing this?
I think that's a good question.
Anyone want to take?
Yeah, well, Angus started to answer and then he froze. I wanted to wait for him to reply. Are you still frozen, Angus?
I think
all right,
it's
well,
frozen.
I'll make a quick comment while we're waiting for him to come back in, and that is, I think it's a great question, Leon. It's right to the heart of something we talk a lot about, which is, it's one thing to show, as many, and this is also relevant for biotechs, right? You can show your POC or you can get an approval, but you're actually getting an ROI on that. Like in this case, I think is, very problematic, right? And that's why, like, our founder of our legacy firm, Ed Saltzman, created that term proof of relevance. Right. And then someone just did a variance at a conference. I think it was a woman, an executive from AstraZeneca, who they were talking about LOE, and instead of kind of LOE, she's called it LOR, loss of relevance, which is a similar idea that, okay, you can get an approval, but there may be no commercial return. So it would not be conceivable even if they had, well, it's complex. I mean, we've certainly seen commercial kills before, right? And not just oncology. The question is, would someone, at this level, would they still try to move it to approval on the hopes that then they could go after other indications and, maybe kind of get some life in that, even if there's not much use in the actual indication in which it was approved originally? I don't know. I guess I'd love to hear your thoughts, and Viraj too.
I honestly don't see any rational. They were trying to get something out of TIGIT. That's what I think. And while working on everything else that they may have, this is big pharma can afford that. We're just gonna go at different paths and see what really comes back. So this is, I think, their strategy to kind of push.
Well, it's almost like it's got its own momentum, right? They've sunk all the cost into it. It's like at this point. Viraj, go ahead.
Yeah, I was just gonna say, I mean, I agree. I don't think I think it's slightly different for biotech versus big pharma. I think for big pharma, the idea is we got our first approval. Yes, it's not going to be the blockbuster, but I don't think we got the foot through the door. Now let's see how we can build on this. For a biotech who is potentially making this their lead program, and this is not relapse refractory single arm 100-patient study based approval. This is going to be a randomized 600-patient study, which is going to be over 4 years. Lots of money, resources needed. So for a biotech who's envisioning that this is going to bring them the value inflection, enable, funding, financing, deal making, then yeah, definitely. And with incremental, just getting approved is not going to make the cut. Not in this environment, because this is not, this is not an environment where just an approval is going to get, the street or funders or investors excited for
Well,
what
I'm
you're
not
going
even
to do
sure it
next.
makes sense.
Yeah.
I mean, it may
Yeah.
make, I
Yeah.
suppose, a large pharma, Roche or otherwise, could make arguments about kind of potential return ultimately, even if there's an irrelevant, emphasize irrelevant approval. But that begs the question of, does it even make sense from the standpoint of, the patients in the clinical trials, et cetera, when they could be devoting those resources, those patients, et cetera, to things that might be much more relevant and promising.
All right, so
Anyway.
we are running behind time, but it sounds like this conversation is getting better and better. Whoever stay here in the audience, good for you. So next slide for Angus.
We got 3/4 left of the audience on with us now, so that's good.
Yeah, that's very good.
So I think this is just basically a slide of the overall survival curve from the tiragolumab study. I show this because I could click it again. The reason why I show this is, Leon, could you click? There's an animation going on. Yeah, the reason why I show this is, like Jeff talked about Harmony-2, like, where is Harmony-2 at right now? You see kind of this curve, at some point is separating, at some point is coming back, and people are starting to wonder, like, because this is also an IO trial, that is also Harmony-2 is also an IO trial, so where are we right now? The question, the answer to that is we basically don't know, and we're not going to see it until probably fall. Next slide, Leon. We're probably going to see it the coming fall. The one thing that I want to elaborate a little bit on this ivonescimab is what Akeso CEO Michelle Xia, she said during the investor call after that kind of readout, was that she was asking like investors, saying that the market interpretation deviated from the core of the issue. She argued that because that Harmony-2 was done mainly to win an approval in China. It was not, it's a small study. It was never designed to really be empowered to show overall survival. So I understand where she's coming from. Basically, like, she was trying to vaccinate investors with this idea that even if it fails, this is not designed to do that. So don't read too much into it. I kind of agree with her.
Love that use of the term vaccinate, Angus. That's great.
Yeah. So I was like, this is to me, I kind of agree, because what are you trying to do with an overall survival readout for Harmony-2? Like, it might give you a immediate stock boost, but in the end, the commercial market is still in the US. You still need Summit to do its US or global phase 3 to really validate the whole thing for the US market. So even if overall survival eventually didn't pan out, you get a kind of a drop in your stock price. But if still Summit can succeed, you can still come back from that. So it's a temporary kind of milestone in there with this overall survival readout from Harmony-2. Do you care about that much? I think Akeso is trying to tell investors, just don't look at that too hard. It's not going to tell you, because we've already got that China approval. Even if you got that overall survival, even if it's statistically significant, you still need that Summit trial to really tell you anything commercially. So right now, but there are obviously different interpretation of her comment as well. Like the bear would say, like, this shows just low confidence in this program, like asking investors not to look at overall survival. Viraj, you want to say something?
Yeah, I guess I was going to say, I mean, I think there is a slightly unrealistic expectation also that people had attached to, based on, what the phase 1/2 had, and, potentially how transformational. Because, I mean, if you think about how IO and even other oncology develop has been, it's always been incremental. Very little has been transformational. And from that perspective, this is there. I mean, you're getting the OS benefit. It's not the 0.7 that typically every, KOL or physician will want, but it's kind of there, right? I mean, it is showing, yes, and it is potentially significant because, it's beyond the 0.8 that, so I mean, it's also associated with the idea that, wow, we thought that, the PG1 VEGF, at least in the phase, PD-L1 positive population where keytruda monotherapy is the star, was going to be transformational. Yes, it didn't reach that point, but I think it's still clinically relevant. And I think it adds value at that, in that patient population. Yes, I mean, the translation of this to the global trial is another thing, and it has to hold up at least at this level. Forget, going beyond and going to that 0.7 or closer to 0.7. But I still think that there is value here. And again, there's probably going to be optimizations to PD-1 VEGFs, which, Jeff and Ting talked about, with a slew of, bispecifics that are out there. And then you're probably going to, build on this. So, I mean, I don't, you're right. It's not a blockbuster win, but I still think it's a pseudo win, in this case.
Yeah, well, as I said at the beginning, even a 22%, is now, of course, if the recrossover happens as Angus showed at the end, and you lose that, survival benefit, that's a different matter. So, yeah,
because VEGF tends to deteriorate when the data become more mature.
That's what people fear right now.
Correct.
Yeah. But I mean, it was funny. I was looking back when, because I knew we were going to end up, this was going to be, at least a hot topic issue. And I was looking back at the history of deal making in IO and the amount of money that people put forward for preclinical assets, right? Over the decade heyday of IO. We're not talking about something that has not even questioning China data. There's just no human data, and people were plunking a lot of money down. So, it's almost like, there's this, and this goes to something I said and Viraj said, it's almost like this overreaction no matter what happens, like over enthusiastic because IO was so down, and over negative when it's not quite as good. Now, hopefully, as I said before, this doesn't become like a Nectar 214 story, but but even if it does for this molecule, there's, all those other molecules, we shouldn't kind of lump them all together. Of course, the worst case scenario is they all end up being like a TIGIT story, and that would be really bad for the industry. But
all right, I think that's all we have. I mean, I will save time. I don't want to share the sentiment too much. Pretty much the sentiment was, I would say the crowds are smaller compared to before. As you remember, during the peak day of IO, when the keynote 189, I believe, presented by Nina Gandhi, I mean, there's probably thousands of people outside the room couldn't get into the room, but I don't say that this time. The crowd definitely smaller, but I would say the interest in China is much stronger than before. We have our own networking events. We have this China Night Summit where Ting and I both went. Super packed, I have to say. We couldn't even get the food. They have to order pizza for us. So they run out of food. It's crazy. I mean, China Night, they give us pizza. That's just tell you how many people showed up. So that's that. And now I do want to save a few minutes for Q&A. People still here. And, there's quite a few questions from the audience. And also thank you, the panelists. They already typed away a few questions. I'll read this one. This is from Tanya. For Angus, do you think considering toxicity of added adjuvant versus options of only adjuvant with others, even if a keytruda get okay in head and neck, actually substantial use is hard to say? You probably can say the question as well.
Yeah, I'm not sure I understand the question. I mean, the one thing that I think is worth thinking about is because adjuvant setting there is a existing adjuvant treatment for head and neck cancer. So there is something that is already established. And so doctors will likely, even though the trial didn't really answer the question of contribution, I think there will be some flexibility in actual use among doctors. Like if you get a major PCR, do I continue to treat patients with keytruda or not? if your patient had already achieved that without kind of adding additional toxicity. So there might be something in the real world setting, some flexibility in adoption, whether they will doctors will continue to use Keytruda.
Yeah,
and
I was just going to add from kind of what I saw reading wires where head and neck experts were commenting, there seemed to be, tremendous enthusiasm for this.
Yeah, just because just how long there hasn't been any new treatment on this. And by the way, there is some comments about like if you were able to reduce this kind of side note, if you were able to reduce kind of some of the recurrence, what does that mean for the medicine, for the companies that are developing in the recurrent metastatic setting, how that will reduce, we'll see kind of the market shrink, how that will evolve for the metastatic setting is something that's also interesting to see.
All right, this last question from our friend, long-term listener, Charlene Liao from Immuno-Onc. And her question, first of all, she made a little bit correction. I cannot confirm. She said they actually, Ateo's TIGIT is actually FC enhanced. With that being said, neither confirm, she asked, how can we reconcile the FC difference among different kind of a TIGIT, right? Some are, FC silent, some are FC enhanced. So, any more colors from you guys?
I think from what I heard is Ateos, yes, it's also the one. Which one was it? I think I misspoke. Was it AstraZeneca or someone? Oh, it's Gilead. Gilead is actually is FC silent. I misspoke. It's not GSK Ateos. Gilead, Arcus, their one is FC silent. So I'm not sure how we can reconcile between the differences. The one based on my understanding from my discussion with Lyric is that the FC competent one, like tiragolumab, is going to come with higher toxicity and at the same time, supposedly higher efficacy. And the Roche one, the tiragolumab, is among the FC enhanced ones, FC competent ones, is kind of on the milder side of toxicities. The other ones, the Beijing, the Merck ones, are even more toxic than that. So this means the toxicity is very high, and with FC silent ones, you kind of have better toxicity profile, which allows you to combine with other agents. That's kind of their promise, like even beyond like the PD-1s, if there are chances, for example, with ADCs, just throwing things out there, to combine with other agents, which may give them a better chance to achieve a better overall regimen profile compared with some of the FC competent regimens. And of course, like I said, there is the GSK Ateos events with their FC enhanced. They're just trying to ramp up efficacy. Let's do high, very strong efficacy to overcome whatever toxicity shortfalls might have.
Yeah, I mean, that would be cool if it worked. I'd like to see an FC enhanced IO agent. Idios has, they have their explanations, including, NK and macrophage engagements, et cetera, as well as killing of Tregs, how that all pans out really when you're in the human.
Yeah, I think it definitely showed. I think it was at ESMO last year where they reported some early data, yes, that very meaningful like phase 2 data in small non-small cell lung cancer data, but investors were kind of were a little bit concerned about the toxicity profile. But so I think eventually we'll just have to see which one kind of have the upper hand in that balance. Do you have does that have higher, stronger efficacy really overcome that toxicity? But again, with lower toxicities, I think at least from Lyric's, my discussion with Lyric was that there may be some better chances for them to do combinations with like PD-1 VEGF or not PD-1 VEGF, like what PD-1 can do.
Yeah. And look, it's sometimes this is not unrelated to the earlier discussion about ADCs, right? I mean, we think, a standard antibody or an FC enhanced or sinus, it's still standard antibody. It should be pretty, predictable, but it's not necessarily, readily predictable. And so, I think, we can hold out hope and see what happens. I mean, right now, what we've seen with a lot of lags up until Immunotep's data was, oh, these are very marginal, but their data looked pretty good that was just released. So something different about that particular antibody? Is that the explanation? Was it the trial design? I don't know. So, but as you said, Angus, proof's in the pudding. So let's see how that, that lag or the TIGIT, et cetera, get across the finish line.
I think I thought Immunotep was just adding, was doing a slightly different approach, right? Immunotep was adding LAG3 versus, I mean, it was doing I thought it was going from the other end compared to the traditional LAG3
Yeah. My point
antibodies,
simply
because
is
I think
not,
that was kind of
that's a fusion protein. Yeah.
Yeah.
But
So-
like OS data from 33 patients, I don't think too much.
Yeah.
2 patients last long, that will change all the picture.
Well, yeah. As we say now, we've gotten more skeptical. 10 years ago, big deal would have been done on 30 patients. Well, done on the bispecific PD-1 VEGFs with, de minimis patients.
Yeah, and also back in TIGIT story. So basically, I think it's been so long, like people, forget to go back, look at what's coming out phase 1, like mono-agent data, So basically, what I'm thinking about, so if, we have to talk about what's the FC makes a difference for those molecules. I don't think there's much hope in this targeted class, So.
Yeah, definitely choppy history, right? I remember I did a deep dive when I was in-house BD and Henlius. I went back for their initial Nature Immunology paper, 2009. Actually, the MOA described in that paper is quite different from what they are talking today. If you go through like the academic publication out of Genentech, actually, their story keep changing. Really feels like academic research. Right, and also Cityscape, if you go back to the phase 2, which kind of set the whole field on fire, there was a controversy because there was no correlation between the, I believe it's a ligand high expression patient and a ligand low expression patient. That was a kind of obscure publication or presentation by one of the PI in one not so famous conference. Actually, that raised a lot of red flag. But that one really has a ratio 0.3 or something. I don't know. I probably now looking back, everyone's smart, right? But at that time, we all believed in it.
Yeah, but that applies to so many things, and I think it's a good point, Leon. Right? I mean, look how many people tried to pursue other biologics to VEGF, and basically you have Avastin, and you got, if you want to count it, Zaltrap, right? Regenerons, which hit more than VEGF-A. And now you've got the biosimilars, and of course they're MTKI small molecules, but that was a long history. I mean, if you looked at the pipeline back then, even we in presentations were saying the same things 10 or 15 years ago, like we've said more recently about PD-1s. Why do we need 30, 40, 50 VEGF, antibodies in the pipeline? And in that case, no one else really made it. Whereas in the case of the PD-1s, we have a bunch of examples of, what are we at now? 12 approvals in the US, something like that.
Right.
So sometimes it's just really hard to judge from kind of the history of, the ups and the downs, kind of whether something will make it. And Avastin is still, still used. I mean, controversies around its use, clearly, depending on indications, but so.
With that being said, just last, probably take everyone to 30 seconds. We're going to conclude. If you really like what we discussed today, think about supporting us as a nonprofit. So we can keep doing this even better. And we're probably going to buy you a drink if you have enough sponsorship in the future. So we're going to be going to ASCO. So there's Asian biotechs in a bar. Literally going to happen again. ASCO Chicago, probably in the same bar. So keep us posted about that. And like I said, the incoming webinar gonna be the recap of ASCO and Bio. First, we're gonna have a networking event in Chicago, and then 2 weeks later, we're gonna have another webinar recap of ASCO and Bio. With that being said, I really, really want to thank the team for you stay all the way until almost 2 a.m. If you guys didn't know, he looks fresh, but just tell you one thing: if you want to be a biotech CEO, you need to have extremely good stamina. I mean, I cannot tell it's 2 a.m. his time. But
Yes,
with that being
thank
said,
you.
also thank you, Viraj. It's approaching to midnight
Pleasure.
for you as well. And
Yeah.
also Jeff, old friend, and Angus, my fellow editors and volunteers from ISWT Community. That's everything we have for now. Thank you, everyone, audience,
Okay.
particularly for showing up today. Thank you. See
Thank
you
you.
next time.
Thank
See you at the
Great-
bar
great
in NASCO.
discussion.
Yes, see you on the bar
Yeah,
NASCO.
see you.
Yeah.
Bye-bye.
Bye.
Bye-bye.
Bye.
Bye. That was great, Leon. Enjoyed it.
Yeah, thank you. Thank you.
Yeah. Yeah, hope to see you at ASCO. Are you doing an event or?
Yes, like I said, we have this brand called Asian Biotechs in a bar. Literally, remember the first one's test run and IO360. That's our first one. That's kind of private.
All right.
This one's public, so the ASCO is going to be a second public one.